Hi everyone, my name is Christoph and I talk about sensation and perception in this lecture. I have done myself perception research on object and faces, so if you're interested in that you can also just Google my name and find it at the TMU webpage and learn more about my own research. So what is the difference between sensation and perception? There are two processes which are actually closely related, and they're actually building up on one or the other. So sensation is the process by which the input of the physical world is received. So we have sensory receptors that receive inputs from the physical world, from our environment, while perception is the process by which the brain selects, organizes, interprets these sensations. So you can see that perception is building up on sensations. What else is important? It's important to note that sensations are rather general. You can say that basically most humans have same sensations. However, perception is something very unique. Every human has a little bit of different perception of the same thing. And why is that? Because every human has an individual history of learning, of memory, of emotion, of expectations, and so on. So in other words, you can also say that top-down mechanisms are forming our perception. So what we have in our brain is influencing how we perceive new things. So sensation, for sensation we have sensory organs, and or more specifically sensory receptors. And we will look at mostly at the eye in this lecture, or basically only at the eye in this lecture. However, it needs to be noted that of course we have a lot of different modalities. We can not only see, we not only have vision, we also have hearing, smelling, tasting, touching. We have body sensation, movement sensation, which is called proprioception. We have pain sensation, temperature sensation. We can tell how we are placed in the room, so we have some sense of balance through the vestibular organ. So there are lots of different sensations, but here in this lecture we focus on vision. So like I mentioned, we focus on vision in this lecture. And here's an example. So you have a tree in your outside world, and this tree is sort of emitting light, and the light is transferred through the lens in our eye onto a layer in the, like an inner layer in our eye, which is called the retina. Now several aspects here. The lens is covered by a pupil, that's the black dot in our eye, and surrounded by an iris. An iris is sort of adjusting how much light will enter the eye. It's sort of comparable to a lens in photography, where you can open the aperture or close the aperture in order to reduce or enhance the amount of light that goes in. What else is important is that the outside world, through this this process of optics, is inverted and presented onto the retina in an inverted fashion. So the tree is upside down. So the question rises, well, if the tree is upside down in our eye, why do we perceive it right side up. And here again is the distinction between sensation and perception. Our sensation shows a tree which is upside down. Our perception shows the tree right side up. Now I mentioned the retina, and the retina is the outermost layer of neurons in the eye. And it does all the processing in this very first step. But interesting here is also that the light enters here in this illustration from the left, and it goes all the way through multiple layers of neurons in order to fall onto the pigment epithelium, the dark layer to the right. So that's the outermost layer in the retina. And from there it is reflected, and then it is captured by receptor cells, which are referred to as rods and cones. So in other words, it has to travel all the way through the retina first, being reflected in the epithelium, and then captured by the rods and cones. From there, the light particles are then transferred or transduced into electric potentials. So this again is cut through the retina. It does not show the pigment epithelium, but it shows the receptor terminals or the receptor cells at the uppermost layer, the RT layer. And it shows how the transduction is taking place here. So the receptor has received a photon, a light particle, which is then converted into an electric potential and sort of connects to different types of cells called horizontal cells, bipolar cells, in a neural network-like structure, and from there it projects into ganglion cells. These ganglion cells are basically the last layer of processing in the retina, from which an optic nerve is leaving the retina. Now, what is important to note here is that the ganglion cells are basically directed more inward the eye than the receptor cells. So the ganglion cells need to leave the eye by penetrating through the layers of the retina at a particular spot, and I will show you later where that exactly happens and what that means for our vision. So this is a photograph of the exact same the retina and the histology of different cells. So what you can see here is that it's actually very easy to keep apart the different cells, there's different cell types in the retina. And you have several layers. You have the RP layer here, which is the pigment epithelium layer. And then you have the PL layer, which is the photoreceptor layers. So you have the rods and the cones here, the two types of receptors. We come to that in more detail in the next slide. And then there are the bipolar and horizontal cells that interconnect and connect to the ganglion cells. And the ganglion cells are here in the second or third highest layer. So that's basically the last layer that has neurons that transfer the input from the photons and transduce this into a neural signal, into an electrical potential. So the ganglion cells are then connected to the optical nerve. well, not connected, but they build the optical nerve, to be correct. I mentioned the rods and the cones. Those are the photoreceptor cells in the retina. And while the cones are responsive for color vision, the rods are responsive for black and white vision. Now, the rods are way more sensitive in terms of light, so you need less light in order to get a response from the rods. However, the cones have higher acuity, so you have higher visual acuity there. And you can also see that there are plenty more rods than there are cones. Interesting is still that we perceive in color, and we not only perceive in color where we look at, where we focus at, but we also perceive in color in the periphery. So again, the difference between sensation and perception here. Now, another aspect which I need to point out a little bit more is there is a point in the retina which is called the fovea. What is the fovea? The fovea contains almost all of the cones. So in the fovea there is color vision. There's no rods in the fovea. The fovea represents the spot with the highest acuity. You have the clearest vision, you have the sharpest vision in the fovea. On the other hand, if you look at the distribution of rods, you can see that rods are distributed all across the retina. Now, the fovea is sort of the 0 point, the 0 degree of rotational angle. So if you focus on something, then you have it in the fovea. And everything else to the periphery can then be described as a change in angle, in degrees. And I mentioned that the optic nerve is leaving the retina. The optic nerve, basically with the ganglion cells, is leaving the retina at 10 to 15 degrees of rotational angle to one side. That depends on which eye you look at. That means at this spot we don't have vision. And what it also means is that you can actually find this spot. You can draw on a piece of paper 2 crosses in a certain distance. Well, it depends how you present this piece of paper in front of you, at which given distance you need to try out, but generally around 10 to 15 degrees of angle should work out, and then you can find this blind spot where you actually don't perceive one of the 2 dots. So you focus on one and you wouldn't perceive the other one because it falls right onto the blind spot. You can also try to Google this, find blind spot, and you will find a description on how to exactly do this and find your blind spot. So the question is now, what happens after sensory processing? So how does neural processing take place? Where does it take place? And how does perception arise? Now, before we can answer that, we need to remind ourselves of several facts. One is that we saw that everything in the outside world is projected onto the retina in an upside down fashion. And that's due to the lens in our eye, which is sort of flipping over everything according to optical laws. Another optical law applies regarding the spatial location of where something is presented in our outside world. Let's take an object which is presented in our left visual field where is it going to fall onto our retina. You can see here in this illustration on the left, in the left eye, you can see this yellow line that indicates that everything in the left visual field will be projected onto the right side of the retina, and everything in the right side of the visual field accordingly will be projected onto the left side of the retina. And this holds true for both eyes. So you have then two information streams, one coming from the left sides of the retina of both eyes and one coming from the right sides of the retina of both eyes. That's indicated here with the color red and blue probably. So the optic nerve basically contains the information from both sides of the retina, and that's always relative to the optic nerve of both of each eye individually, of course. The optic chiasma is then the point where the information from the optic nerve splits, and the two sides of the two similar sides of the retina from both eyes merge into an optic tract. So again, you have the optic chiasma, and you can see that at the optic chiasma the optic nerves are split according to which side of the retina the information comes from, and that holds true for both eyes. And then it merges into an optic tract according to where the information originated from regarding left or right side of the retina. You can see that in the color red and blue. And this optic tract leads then to the so-called lateral geniculate nucleus, or here lateral geniculate body, the LGN, which is part of the thalamus. And the thalamus basically acts as a relay center for visual processing. And then from the LGN, there is a geniculocalcrine tract that leads into the primary visual area of our visual cortex, particularly into V1. So what this design principle also implies is that the primary visual areas receive the images of the contralateral visual fields from both eyes, which is illustrated here in the color. You can trace that back. Another important concept which we need to know is receptive fields. What are receptive fields? Receptive fields are contained of photoreceptors. And these photoreceptors, like we have seen through horizontal cells, bipolar cells, merge into a ganglion. Now, photoreceptors are not equally sensitive to light, but some of them are turned on fully by light, others are inhibited by light. And together they build a receptive field. So typically receptive fields have on centers and off surrounds. And if you see here in this illustration on the right, you can see that several scenarios of stimulation will create different activity level in the ganglion cell as well as in the bipolar cell. And you can see that the maximum activity is achieved when the on-center is fully illuminated. It is partly achieved if the on-center is illuminated by a spotlight, like the upper illustration. And then you have all the scenarios where it's not maximally activated, like a spotlight going to the surround will not activate receptive field, that ganglion cell. The concept of receptive fields has also been further investigated by Hubel and Wiesel in 1968. And Hubel and Wiesel received the Nobel Prize in physiology and medicine for their work. What they did is they presented a stimulus on a screen and they recorded from brain cells in the cat. What does that mean? They put electrodes into the brain of the cat that recorded the extracellular responses from neurons. Now, originally they wanted to find what kind of stimulation is triggering a response. And then by accident they found out that line stimuli are triggering a response as opposed to what they thought, like spotlights or shapes or whatever. So line stimuli are triggering a response in the V1 cortex, in the first visual area in the cat brain. They also figured out that not every line is triggering a response in every neuron, but neurons are tuned according to the orientation of the lines. As you can see here, you have a stimulus orientation in this graph here at the lower left. You have a stimulus orientation in degree on the x-axis, and you have a spike response on the y-axis. So you can see that the spike response increased systematically the closer you go to a 0 degree of rotational angle. That's now one type of selectivity, but there have been all different types of selectivity. And interesting is also that the orientation selectivity is systematically arranged, topographically arranged in V1 cortex in the cat and also in other mammal species. Now, what's the consequence of having receptive fields? Why is it important for the analysis of visual inputs to our brain? Imagine we have receptive fields, not only center surround receptive fields, but we have these blob-like or line-like structures like receptive fields, like we just saw before by Hubel and Wiesel, that selectively respond to a certain orientation of lines or blobs. Now this is basically the key element to object recognition. Of course, there is more to object recognition than this, but this is the first initial step of a potential processing of an object. Let's assume we look at a tree. How can we recognize this tree? Well, we can recognize it by the fact that the tree consists of a strong vertical line and then branches going out in certain angles. Now this strong vertical line, the stem of the tree, can be described by multiple neurons that sort of align in this vertical dimension, and all of them fire most for a vertical line. So this would indicate here there is a stem of a tree. So when we look at the cortical processing, we might get the impression that the brain is extracting features from the visual stimulus, and it actually does that. There are so many receptive fields of cells in V1 that are only selective to bars. They're tuned to finding bars, edges, corner pieces with a specific orientation and direction, even direction of movement for dynamic representations. So that's really the idea, that at this very early level we extract features from the visual stimulus in our outside world. So that's also an important aspect in terms of object recognition. So how do we recognize a tree? The visual system actually builds up the representation of that tree by detecting features like these lines that make up the tree's contour. And the pattern of the response of a number of such features might then indeed indicate a particular object, like here a tree. So the question arises, does our brain use visual features to detect an object? We saw that at V1 level there are receptive fields for bars, edges, corners, in specific orientations, and also in direction of movements. The assumption is therefore that at some level in higher levels of visual processing, there might be cells that respond selectively for particular objects which we perceive. Now, nowadays we have an answer to this, and the answer is yes, there is. In 1959, a researcher named Letwin did a study. He recorded from the visual system of the frog, and he found that there is some sort of fly detector implemented in the ganglion cells in the frog. What does that mean, a fly detector? He described it as a movement-gated dark convex boundary detector. What he means is that a dark spot moves through the visual field of the frog and activates ganglion cells one by one. He was one of the first, or the first, who suggested this. He suggested that cortical processing is feature processing in terms of, vision. And nowadays we have a lot of studies showing exactly this. However, at the time, he was laughed at conferences. He had problems to publish his work. So the study by Bruce and colleagues in 1981 was among the first to show that at higher levels of visual processing, we can find cells that selectively respond to particular objects. He showed that there are cells responding to line drawings of monkeys, of humans, of hands, of cartoon faces, of pretty much, all type of faces. Nowadays, we also have studies showing that there are cells responding to any particular object. So the question is, do we have sort of, in quotes, grandmother cells? Or do we have a yellow Volkswagen cell? Do we have a yellow cell and a different type of cell that responds to a car? And together they indicate that it's a yellow Volkswagen. So that's the debate now. Is it a combination of features that activate simultaneously? Or is it sort of the ultimate stage where we have a cell that is responding to any given object in the world? So, because many different areas in our brain, especially visual areas, have been identified anatomically and physiologically. There is now a tendency to sort of attribute specific functions or functional aspects onto different areas or regions. So for example, take the visual area V4. It has been identified as an area that is responsive to color. So it must be the place where predominantly color is being processed. Similarly, there's an area called MT, which is in V5. So V5 has been shown responsive to moving stimuli. So is that the area where movement is being processed? Is it the motion processing center of our brain? I think that would be a bit too simplistic to say. Definitely there is some sort of processing of color and motion and depth and binocularity, and we have seen orientation going on in separate models. However, there's a strong connectivity between cortical areas. The processing of vision is quite complex, and particular areas to be assigned to only one type of processing, like color only or motion only, is a bit too simplistic. However, what has been shown is that particular damage in cortical regions, in these specific cortical regions, can lead to specific losses in visual perception that are related to these different features like movement, color, depth, etc. So far we have been talking about primary visual areas like V1 to V4, and those are located in the occipital lobe or in the striate cortex, how it's also referred to. Now in 1982, Leslie Ungerleider, who is a famous researcher in this field, has identified two processing streams. One that goes from the striate cortex into in the occipital lobe to the parietal lobe, and another one that goes into the temporal lobe. So you can see here the parietal lobe would be the red color and the temporal lobe would be the light green color. And the study refers to these two streams as the what pathway as opposed to the where pathway. The what pathway refers to the temporal lobe, so it basically is dealing with what is it, what I perceive, so it's sort of an identity processing, whereas the parietal lobe, the where pathway, is dealing with the fact where is it located, where is the object located. So that's the where question which is being dealt with in the parietal lobe. So Ungerleider, Mishkin in their study have trained monkeys to perform two tasks. You can see A and B. In one task, which is called the object discrimination task, the monkeys were presented with an unfamiliar object, and they had to and they were then presented with another object, and they had to identify which one is the new one in order to indicate that they recognize the other one, the previous one. And then they were rewarded if they solved the task correctly. So the monkeys learned this relatively quickly. And in a second task, they, which is referred to as a landmark discrimination task, the monkeys are placed in front of a table that had two what they called food wells. So you could open them and then you could withdraw food if there was some food reward inside. And a cylinder was placed next to one of the food wells. So if the monkey was choosing the food well next to the cylinder, it gets a reward. And also here the monkeys learn to do that relatively quickly. So they associated that if an object is placed next to the food well, then there must be a reward in the food well. Now, after training, these monkeys have received a bilateral dissection of an area in the temporal lobe or in the posterior parietal lobe. So that's indicated here with the dark shading here in the outline of the monkey brain in A and B. So they have been removed, these certain parts, and the monkeys then were not able to solve these problems anymore. So the monkeys who have temporal lobe lesions are no longer able to perform the object discrimination task, and the objects that had the monkeys that had a parietal lesion can no longer perform the landmark discrimination task. And this is a clear indicator of the distinction of where and what, or what in A, object discrimination, versus where is the object shown in B. In the study by Ungeleit and Mishkin, the lesions were sort of artificially induced in the monkey brains. However, it also occurs naturally. And of course, it also occurs in human brains. And here's an example of what can happen if things go wrong. And again, it's important to note that this has nothing to do with acuity or visual field or scanning or language, memory or intellect. So these people are perfectly fine in all these regards. What they have is a lesion in this higher level of visual processing that leads to certain deficits, and one of them is visual agnosia, which is the impairment of recognizing visually presented objects. And what you can see here is that a subject that's suffering from visual agnosia has received sketches, outlines of, or drawings of a key, a bird, a pig, and a train. I had to copy that. So the drawings that are marked with an asterisk are the ones done by the subject, and you can see how hard it is for a subject suffering from visual agnosia to copy one-to-one an image. So you can see that certain details are not exactly matching up perfectly. So it's a very, very difficult task. So here you can see a different study where the subject was asked to name presented objects, and you can see that most drawings don't match up with the naming. What is also interesting is you can see that there's certain similarity, visual similarity, between what is believed to be shown here and what is actually shown here, like the pretzel and the snake in B, or the lamp and the mushroom also in B. So it seems like the subject is able to pick up this sort of general shape, but then the detection, the identification of the object is not possible. Another form of a deficit is prosopagnosia. Prosopagnosics cannot recognize faces, even of close friends and relatives. However, they can identify these people by their voice or by the movement. So, prosopagnostics can actually deal with this situation quite well. They find other ways of identification of a person, because it's a multimodal task. It's not only restricted to a face if you need to identify a person. So prosopagnosia is also called face blindness, and we cannot imagine how that looks like, but the picture shown to the right is a rough idea of how it might look like. So prosopagnostics cannot recognize familiar faces. They cannot see. They cannot even recognize their own spouse, for example, or their own family members, just based on the face. To some extent, they can recognize particular features, but they cannot recognize the same set of features repeatedly. In order to point out a little bit more the distinction between what versus where streams, let's focus on another damage in the where stream, which is called contra or unilateral hemi-neglect. So in these conditions, patients have a damage in the parietal lobe that leads to an unawareness of what is going on in the visual field contralateral to the side of the brain that has that damage. This might result in behavior like patients eat only half of the foods in a plate and leave the other one untouched, or they only dress one side of the body and leave the other one untouched. Men only shave half of their face and leave the other one untouched. And and these patients mostly are not aware of they are not conscious of the perceptions in this hemifield. And here in this drawing, you can see that someone was asked to draw or paint flag of Canada, a smiley face, a tree, an apple, a flower, and a house. And that's the way it turned out. So now you can also infer from this painting or drawings where the lesion must have been. So here's another example of a study that looked at the contralateral or unilateral hemi-neglect. And you can see that copying on the left side resulted basically in only attention towards one side of the model, which was supposed to be copied. And it's particularly obvious when you look at the clock. And in spontaneous drawing, it basically produced images which are focusing only on one side of the representation. However, some parts actually are present in this drawing in the human face. You can see there is an ear on the left side even. There's no eye on the left side, but on the right side. So up to this point, we have seen the processes involved in sensation, and we have seen how the brain is processing these sensations and creates perceptions out of it. But now perception is interesting also from a different point of view, and that's perceptual illusions. And the perceptual illusion, according to the Oxford Dictionary, is the misinterpretation of a real external sensory experience, the action of deceiving, especially by appearances, an instance of the sense perception of an external object, suggesting a false belief as to its nature. And what that means, you will see now with a series of different illusions, which we talk about in the following slides, and we talk about what that means regarding sensation versus perception. So the first illusion we talk about is the Miller-Lire illusion, which is named after the inventors of this illusion. And it goes that what we perceive is that the vertical main lines of these two shapes are perceived as of different length, although they are of same length. So you can go with a ruler and compare, but they are definitely of same length. Now the explanation to the phenomenon, so why we perceive the left one here in this image as longer as opposed to the right one, is not so clear, but it's being speculated with regard to geometrical perception of these lines. So one explanation is that the visual system learns that the angles pointing inward configuration would correspond to a rectilinear object, such as the convex corner of a room, which normally is close to the observer, and the angles that are outward directed, so the angles out configuration, would correspond to an object which is far away, like such as the concave corner of a room or a building. So that's one explanation for this. What is important again in this lecture is that you realize that sensation and perception are different. So what the sensation tells you here is that all these midlines are exactly of the same length. What your perception tells you is that the left one is perceived longer than the right one. So what we see here are bistable figures, and maybe the most known is the upper left one, the cube that's called the Necker cube. And the idea is that there are basically 2 points of view at which you can look at these figures, and you can flip between the two. So try and look at these figures and try to see if you can see them both and flip between them. The one in the middle is called the Rubin vase. So also try to see there if you can see the faces, and then flip to the vase and flip back to the faces. So when you look at these types of figures, they probably spontaneously flip back and forth between the two percepts. Usually, you have one interpretation which stays a little bit more prominent, but then it might flip back to the other one. You might have your tendencies, your preferences of seeing one above the other. Like in the Necker cube, you normally see the one pointing down and not necessarily the one pointing up at first glance. So the question remains, why does your brain actually do that? And why does it stick to one or the other and flips back and forth? And the answer is that maybe, these figures are not clear. They don't provide enough information for the brain to be clear in terms of interpretation. So there is no correct solution, and the brain tries to interpret both ways, or all the ways there are, and then flips back and forth between them. And you can see that there are some very simple versions like the Necker cube or the Rubin's vase, but there are also more complex versions like actual paintings or drawings, like the one in the lower left which says Kaninchen and Ente. Kaninchen und Ente means the rabbit and the duck, so you can flip back and forth between the rabbit and the duck. Try if you can see that. And then also the one to the right, which is a young lady from the back and an old lady from the front. Now, why are bistable images so interesting or fascinating even? I think it shows again the main message also of this lecture, that illusions show that perceiving is more than just the direct experience of a visual input, like sensation. It is more than sensation, so it's a projection into the brain, and the brain basically tries to make sense of what it perceives. So the brain tries to make sense of the world. And that's, again, the difference, sensation versus perception. Now, the effect of afterimage is also quite interesting one. You can actually try this. You can try to stare at this American flag for a long time, for 20-30 seconds, without moving your eyes around. You just focus on the middle of it and look at it. And then you look at the white wall, and what happens is you actually see the American flag in its original colors. Now, of course, that's again the idea that sensation is not equal to perception, but we can also try to explain this effect now. So what happens here actually is that when we look at something very strongly, we focus on something very strongly, like the stimulus, and light-sensitive photoreceptors, like we discussed before, are stimulated in the retina to incoming light. And we also need to know that there are different types of cones. There are short waves, mid-wave, and long-wave cones responding to different frequencies of light. And some of them are more active than others due to the colors you can see in this image. However, if you look at them now strong over a long time, then these photoreceptors become sort of desensitized or fatigued, so they become tired. And the photopigment which is released is bleaching out, and through these strong and constant stimulation. So what happens then is that the desensitization is strongest for the cells viewing the brightest part of the figure, but it's weaker for the cells that look at the darker parts of the figure. So when the screen becomes white, or when you look at the white wall, the least depleted cells respond the strongest. So more than the neighbor cells, for example, producing the brightest part of the after image and producing sort of the opposite colors than what you perceive in this actual stimulation. So this next illusion is the cafe wall illusion, and there are multiple versions of that. And you can see that the main point is here that you have black and white tiles here arranged in a horizontal fashion, and then there's a subdivision as a tiny little line that goes in between the tiles. And this line is perceived as either ascending or descending in the left version of the illusion, or as a wave-like line in the right version of the illusion. Now this line is critical, and what is important is that the degree to which the tiles align here in a vertical manner determines the amount of effect you see, so the amount of ascending or descending lines or wavy lines. And the explanation, according to Gregory and Hart, is they suggest a border locking approach to describe why this illusion works. Their idea basically is that highly contrasting shapes or shades help your brain lock in the location of different objects. So if the rows of black and white tiles were right on top of each other with no border, our brains could more easily see where the corners line up or don't. So with a narrow gray line between the rows, our brains can't quite lock down where things are happening in relation to each other. So this is one possible explanation, but I think that's not the end of the story. So most of these illusions are probably very complex, and interpretations or explanations of them are not always known and probably not always so straightforward. So again, this is an example that our perception is way more than our sensation. And here we have another cafe wall illusion, here in an invert, or in a fashion where the illusion takes place in a vertical dimension, and the alignment of shapes that determine the effect is in a horizontal dimension. And here also what the inventors did is they brought in different colors in order to enhance or decrease the effect. So here we have the herring illusion, and this is a very old illusion from 1861. It was invented by Ewald Hering, a German physiologist, and it basically shows that if you have two parallel straight lines in front of a radial background, what happens is that these lines tend to bend outwards. And the question is, again, why does that happen? And there are multiple ideas and hypotheses and theories trying to account for this. Hering himself postulated that the illusion is sort of an overestimation of the angle which is made at the points of intersection with the background. And then from there, others try to come up with other explanations, that the angle is an overestimation as a result from lateral inhibition in the visual cortex, or it is a bias inherent in extrapolating 3D angles information from a 2D projection, which is generally what happens if you perceive a 3D image in the world. You have a 2D projection in your representation. So altogether, there is no actual explanation for the Hering illusion so far. This illusion is called the spinning dancer illusion, and the idea is that the girl that is spinning around here, the dancer, can spin around clockwise or counterclockwise. The illusion most likely arises from the fact that there is no real information about the depth. So we do not have any visual cues regarding the depth of the image while the dancer is rotating around this axis. There has been found a correlate using functional magnetic resonance imaging in the right parietal cortex, which generally reflects the notion that that information in space is processed in parietal cortex. So this is an example of color constancy. And color constancy is subjective constancy of color. It's a typical feature of color perception in human brains, which sort of ensures that the perceived colors of an object remain relatively constant and do not vary too much under changing illumination conditions. So this is also an example how, we know that strawberries are red. We know that it's in our memory. The association to strawberry is red. So even though we do not have so much color information here, we perceive it as a very strong red. Well, this is the topic which we discussed before, the blind spot. And the blind spot is a tiny little spot in the back of the eye, in the retina, where the ganglion cells are leaving the retina through the optic nerve. So at this point you have no perception, and here with this arrangement of the shape and the cross, you can actually test it. So normally you have a distance between 12 to 14 centimeters between the center of the shape arrangement there and then the cross. And if you, in this arrangement, you need to close your right eye and focus the cross, and then try to see, by varying the distance, if the shape to the left would change in the center, like you would not see the center of the shape anymore, it would disappear, and then basically you have hit the blind spot. Again, if you want to try this, there are many versions on the internet available, which might be a little bit more straightforward. You can try out a lot of them. Another perceptual fill-in is the fact that we tend to perceive shapes, lines, borders where they are not. We also try to cluster objects, even though from the sensation point of view they are not clustered. So let's take different examples. Here you can see in the upper left there is a triangle in the middle, a white triangle. Even though there is no triangle, we perceive a triangle. The WWF panda in the lower left is an arrangement of white and black blobs, but we perceive it as a panda. So we sort of complete this into an object. The lower middle example marked with the letter C shows nicely how we perceive a sphere, even though there is no sphere here. D shows some sort of snake going through the water, for example. So you see what that means. There are certain laws according to which our perception works, and we will go into more detail with these laws. So these are called the Gestalt laws or Gestalt principles, and yeah. So a further perceptual fill-in illusion is the Fraser spiral, and this is quite an interesting illusion. You perceive a spiral, however, if you look closely, you can see that it is not a spiral. It is circles that leave the impression that they are a continuous spiral. So you can go and trace individually these one outline of a circle, and you will see that it's actually a circle. So here you can see the overlay of the circles onto the perceived spiral. Now this next example is very interesting. So you have a rotating arrangement of blue crosses. In the middle you have a fixation dot that blinks green, and if you focus this fixation dot in the middle, what you realize is that the three yellow dots in the periphery disappear one by one, sometimes all of them simultaneously, sometimes only some of them, and then they reappear again. Now let me assure you that in this presentation, these yellow dots are always present. So it's your perception that makes them disappear. The explanation to this phenomenon is not known, but probably something like motion blur suppression that is related to this motion-induced blindness. And if that's the case, then New and Scholl concluded that rather than being a failure of visual processing, motion-induced blindness may be a functional product of the visual system's attempt to separate distal stimuli from artifacts of damage to the visual system itself. And this is actually quite an interesting idea. If that's the case, then there's an active process going on that the brain and the perception as the product of the brain is trying to fix things which are not perfect, which it believes are not perfect. So in this example, we have the Leaning Tower of Pisa. And what an observer normally observes is that the right Leaning Tower of Pisa is a little bit more tilted to the right, so in a more precarious position than the left one. However, they are the exact same images. Now, why do we perceive the right one as being more leaning to the right than the left one? There is an explanation for this, and it's has to do, with the perspective that we take when we look at a tall building. So imagine you have two tall towers, and you take a photo from below. If the two tall towers are endlessly tall, eventually you will perceive them as touching each other at the tip. Now here the situation is different. You have a leaning tower and the same leaning tower adjacent to it. So you have a series of leaning towers. And probably our perceptual system interprets this as sort of a sum that while one leaning tower is leaning, the other one must be leaning more, the third one must be leaning more, and so on. So our visual system must assume that they must be sort of diverging as they recede from the view. And this is what we then perceive. And this is exactly the opposite. So it sort of works into both directions. Now the next illusion is a visual illusion regarding size contrast, and it's called the Ebbinghaus illusion. So the Ebbinghaus illusion goes the following way. There are two orange colored circles, and they are of same size. However, these circles are surrounded by gray circles of larger or smaller sizes, making the relative size of these orange circles either smaller or larger. However, if we test this, they are exactly of exact identical sizes. The Ebbinghaus illusion also has played an important role in the debate over the existence of separate pathways for perception and action in the brain, according to the two-stream hypothesis which we introduced before. Since the Ebbinghaus illusion only works for visual perception but not for action, so this is still an ongoing debate, and even today many people are still working on the Ebbinghaus illusion and the interpretation, explanation of it. Similarly, there's a geometrical optical illusion called the Ponzo illusion after the Italian psychologist Mario Ponzo. And the idea here is that you would overestimate the length of the yellow bar in the upper part of the image as opposed to the length of the lower part of the image, although the two bars are exactly of same length. Now the idea here is that there's sort of a 3D illustration implied. Also probably we would interpret this image as an ladder going up or as railroad tracks going into the distance. And by this sort of implication, we probably would interpret the upper line as longer. Since it is further away, it must be longer than the lower line. Similarly, the illusion on the right, where the two characters are of exact same size, however, due to the visual cues that are given in terms of depth and space, the one that is further inside the tunnel appears larger because it must be further away and therefore it must be larger according to our perception. So this illusion basically is using the extended perspective of this image, and you would probably think that the distance from A to B is larger than the distance from C to D. However, if you go and measure it with a ruler, you can see that they are exactly the same. So again, here our perception is tricking us a little bit into believing that CD is shorter than AB. So I showed you a lot of different illusions and also effects on how our brain is creating a perception which actually does not correspond with what is out there in reality with our sensation. Why does the brain do that? Well, the answer is simple. There is 99% of the cases where such cheap tricks work, and in most of the cases they work, and they are coherent with our assumptions and our beliefs and so on. And then it saves a lot of time and energy if the brain does it this way, rather than if the brain does need to compute that each time anew. So it's well worth it. And you need to remember that neurons are actually quite dumb. Neurons themselves do not code much. They are relatively dumb. It's the entire cell assemblies and areas and the whole brain in its interaction that creates this phenomenon which we call perception. Generally, there are a certain amount of Gestalt principles, I have mentioned them before, that help us humans to perceive our world. And we can go through them one by one. There are a lot more than the six which are introduced here: proximity, similarity, closeness, symmetry, common fate, and continuity. The Gestalt principles are the result of a trend in a direction in psychology that was called the Gestalt psychology. You can also Google this, you will find a lot of information about the Gestalt psychology. This is predominantly a direction from the German psychology, and Gestalt actually implies the shape, the holistic appearance of a thing. So the first Gestalt principle is proximity. The idea of proximity is very simple. You have on the left 6 vertical lines, and on the right you have 6 horizontal lines. Why now don't you have 9 horizontal lines on the left and 9 vertical lines on the right? The answer is proximity. So whatever is close to each other belongs together. And here 9 dots are close to each other, so they belong into one category, into one unit. So you have 6 vertical lines on the left and 6 horizontal lines on the right. Another Gestalt principle or Gestalt law is similarity. Everything which is of similar kind belongs together. So here you probably would not assume that this is a vertical arrangement of dots, but it's a horizontal arrangement because these dots have the same colors. And then we have the Gestalt principle called closure. And closure actually we have seen before when we saw the picture of the WWF panda, the symbol of WWF. There we have something like closure, where an arrangement of patches, white and black patches, are sort of combined into a holistic appearance, into an object. That's called closure. And here we have simpler objects of a circle and a rectangle. And I call them even the circle and the rectangle. I would not say an assembly of circular lines with holes in between, and an assembly of lines that build corners that shape like a rectangle. So for me, that's these shapes are circles and rectangles. And that again implies that what I do in my brain is I use a function closure that sort of tells me that these individual lines belong together into one object. Another Gestalt law or Gestalt principle is symmetry. We like to have symmetric objects, and probably this tendency, this preference for symmetric objects comes from nature, because in nature a lot of things are to some degree symmetrical, at least from certain perspectives of view. So that's an important other criteria, and symmetry actually is quite a common characteristic if you pay attention to all different brand labels, company labels. These labels are often designed in a symmetrical way. So next time you go out and are confronted with whatever labels, pay attention to it, and you will see there are a lot of Gestalt principles in these labels. So another Gestalt principle is continuity. We normally tend to believe that whatever is aligned according to a particular dimension, let's say here in a line, is belonging together. Like the lower left picture here, we have two lines, and these two lines sort of classify the dots into two categories. If you take the lower right image, it will be a little bit more tricky. So here you have continuity on one hand, and you have similarity regarding the color on the other hand. So here you have a competition between continuity and similarity, and it's up to you to decide what works for you. Common fate basically is the idea that whatever moves into the same direction sort of belongs together. So let me summarize this lecture and give you some final remarks. We saw throughout the lecture that there's a differentiation between sensation and perception. I think I made this very, very clear. Sensation is the basis for perception, but perception is much more than what is actually out there. This also gives us a new glance at how we need to perceive other people. Everyone among us has a different perception on the same thing. So if you don't agree with someone, then maybe, you have different perceptions. And this also is sort of an, the idea that, well, take it as something very subjective. Everyone is different and everyone has a different perception. And this is also shown here at this very basic level of visual perception. Then why does the brain do all that? Why does the brain add so much by itself where there is not in reality? Well, it's the same what we talked before. It helps and it works in most cases, and it helps to save resources and time and energy. Energy is a critical element. If you need to compute everything all the time constantly, then our brain probably would need much, much more energy. If, however, you can rely on some memory stored information, on associations of expectations and emotions, then you can compute the same within fewer time and with fewer energy. Also, what is important is that what we saw here is that through visual illusions, for example, you can actually tell quite a lot about how neural processing is going on. And that's also one more message, that for doing a good psychological experiment, you don't necessarily need to see what's going on in the brain at the cellular level, at the level of areas and cell assemblies and cell patches. You can probably infer a lot of it by having a very smart behavioral experiment, or by having an interesting illusion that points out, differences in neuronal processing at different levels. So, it's not always about the resources that one has, but more about, what you can generate, what you can come up with in terms of good experiments, and that with very, very simple means. And then again, we only talked about visual perception, actually visual sensation, visual perception, but there is more. There is also acoustic sensation and acoustic perception. The same principles apply there. And there are many, many more modalities. And then there's an integration of all these modalities. So, the same rules apply at very different levels. So this is basically the take-home message, and I hope I could somehow, explain you all this in an understandable manner. Okay, thank you very much for your attention. Bye-bye.